Hello, we are SOTP 5, a group that aims to educate about the genetic condition Tay Sachs disease. Finding out that your child has Tay Sachs is undoubtedly shocking and difficult to come to terms with. I'm sure you're hearing a lot of jargon and terminology that might not all make sense to you. Therefore, we aim to specifically help you understand what Tay Sachs disease is and what it means for your child's future by answering the questions: how Tay-Sachs occurred, what are the causes and symptoms, how did my child get this disease, and what is the outcome for my child. In this video, we start with explaining the mechanism of Tay-Sachs disease, followed by the review of the symptoms, diagnosis, inheritance, and a summary of the research being done. At the end, we also included some resources to help support you as parents in this trying time. Tay Sachs is a lethal neurodegenerative disorder which is caused due to deficiency in hexosaminidase enzyme. This further leads to accumulation of GM2 gangliosides in the lysosomes of central nervous system. Here, lysosomes are responsible for digesting nutrients. So the accumulation and the undegradation in neurons eventually leads to Tay Sachs disorder and thus leads to death. It is an autosomal recessive disorder in which the genetic trait can be inherited from parent to child. Tay Sachs is very common in Ashkenazi Jews, French Canadians, Cajuns, Old Order Amish, and Irish ancestry.
What is HexA? HexA is a gene on chromosome 15. So a gene is a little section of the chromosome, such as this section that I have circled. This HexA gene will code for the alpha subunit of the enzyme beta-hexosaminidase. That's this here. So this structure that I'm showing is a visual illustration of what the protein looks like when the chain of amino acids folds into the protein. But for simplicity, we'll be looking at this model. Now, Tay Sachs is caused by a mutation in the HexA gene, that's this, that results in a defective alpha subunit being formed. The severity of Tay Sachs disease is inversely proportional to the functionality of the beta-hexosaminidase that is present. When I say the functionality of beta-hexosaminidase, I mean that they measure relative to that of someone who does not have Tay Sachs disease. Therefore, someone with Tay Sachs disease has a lower functionality than someone who does not. With increasing beta-hexosaminidase function, you are closer to the normal person, and if there is decreasing beta-hexosaminidase function, you will have a more severe version of Tay Sachs disease. Therefore, infantile will be worse than juvenile, and juvenile will be worse than late onset Tay Sachs disease. Another way of thinking about it is that a person with infantile Tay Sachs disease will have a much lower beta-hexosaminidase function than someone with juvenile Tay Sachs disease. So what types of mutations can there be in this HexA gene? Normally, this is what you get from the HexA gene. You get this protein. However, there can be different mutations that result in, for example, the substitution of one amino acid in that protein, or the addition or elimination of amino acids. So let's consider this example. This is one mutation that might occur in the HexA gene, and it results in a protein that is truncated. That is, part of the protein is missing. Therefore, this will probably have very reduced function. Another mutation, like this one, might result in a protein that has a different shape. See how this shape is different from the original? And this can have various impacts on the cell itself. What impact does mutated beta-hexosaminidase protein have? Beta-hexosaminidase is needed for the breakdown of a lipid called GM2 ganglioside. So what it does is that this enzyme will take the GM2 ganglioside and break it down. And this all occurs in the lysosome. So this is where you find the beta-hexosaminidase. Normally, what happens in a cell is that you have an amino acid chain, and this is formed in the ribosome here. This is the ribosome. And this amino acid chain will go through many, many processes to form the alpha subunit. And this alpha subunit will go into the lysosome, combine with the beta subunit, and form your fully fledged beta hexosaminidase protein that can break down GM2 ganglioside. However, what can also happen sometimes is if you have a mutation in that HexA gene, there will not be the formation of the normal alpha subunit. Instead, it will form a mutated alpha subunit, and this mutated alpha subunit might not be able to enter the lysosome, or lower concentrations of the alpha subunit might be able to enter the lysosome. And this is how you get decreased activity of beta hexosaminidase. Now, what you want to remember is that less than 10% of beta hexosaminidase activity means that you will have TSD. So let's say that you had exactly 9% of the normal beta hexosaminidase activity. You would be less functional than someone who had, for example, 10% of beta hexosaminidase activity. And if I have less than 10% of beta hexosaminidase activity, then I have Tay Sachs disease. If beta hexosaminidase is not functional, then GM2 ganglioside will accumulate in the cells. That means that you will have the formation of these vacuoles that are filled with the GM2 ganglioside in the cell. Since neurons have the highest GM2 concentration when compared to other bodily cells, the neurons are impacted more than any other bodily cell by this accumulation of GM2 ganglioside. And when there's accumulation of GM2, it can result in neuronal death. That is, death of the cell. And this can have impacts on the human body. So you'll see physical impacts such as a decrease in movement, loss of muscle coordination, loss of speech and balance, and then you can also have such conditions as seizures. In summary, mutations can cause premature termination and ineffective intracellular transport. Inheritance of copies of mutated HexA gene causes deficiency in the enzyme's activity, and this leads to failure to break down a GM2 ganglioside in the nerve cells, and that results in the disruption of the CNS, or central nervous system.
The symptoms of Tay Sachs is based on the onset of the disease, with the most severe symptoms present in the infantile onset of the disease. This is because in infantile onset, in which the symptoms appear within 3 to 6 months of birth, the child has very low levels of HexA enzyme present. This leads to symptoms such as reduced motor ability, muscle spasms, muscle twitching, increased sensitivity to sound, and abnormal eye movements. Another symptom that's present in the infantile onset and other onsets as well is the cherry red spot found behind the eyes. The cherry red spot is apparent in 90% of Tay Sachs cases, which means it can be a source of diagnosis as well. In juvenile Tay Sachs, where symptoms appear within 3 to 10 years, moderate HexA enzyme levels are present in patients. This leads to less severe symptoms such as muscle weakness, muscle spasms, and hypotension, which means low blood pressure. In late onset, where symptoms are presented in age 11 to 20 years, high functional enzyme levels are present. This means that the symptoms are less severe. However, symptoms such as central nervous system degeneration, less movement, less responsiveness, and lower blood pressure is still observed. How is Tay Sachs diagnosed? In a blood test screening, enzyme levels in blood is assessed using an enzyme assay, and reduced or absent levels of HexA is an indicator of Tay Sachs. The presence of a cherry red spot in the back of the eyes is also a symptom of Tay Sachs, and if detected during an eye exam can also be an indicator of Tay Sachs disease. However, the proper way of diagnosing Tay Sachs disease is a molecular genetic testing to detect mutations in the HexA gene that are responsible for Tay Sachs. It should be noted that the molecular genetic testing is only available in certain labs. Tay Sachs can also be diagnosed prenatally through the use of screening such as amniocentesis, which can be used to detect if the developing fetus has a chance of developing Tay Sachs in the future.
We talked about Tay Sachs disease being caused by a recessive allele of the gene. So let's see what this means for the transmittance of the disease. Here is a pedigree chart where both the mother and the father are carriers of Tay Sachs. In this case, there can be three possible outcomes of for the child. When both the parents passes on a normal gene to the child, that means the child is normal with no disease. Secondly, if one of the parent passes on a normal gene, but the other one does not, that means that the child will be a carrier with no disease. However, if both the parents passes on the HexA-associated mutation to the next generation, that means that the child will be affected with Tay Sachs disease. This suggests that Tay Sachs is an autosomal recessive disorder, meaning that both the parents needs to be the carrier in order to pass on the mutation, the HexA gene mutation to their children. Unfortunately, Tay Sachs is fatal at the moment, but while there are no treatments, we can take certain preventative measures to lower the probability of having a child with Tay Sachs. One of the preventative measures is in vitro fertilization. In vitro fertilization is a technique where a mother's ovum and a father's sperm is fertilized in a lab. The genetic testing for Tay Sachs is done on the embryo in order to check whether the embryo is affected with Tay Sachs or not. After, only the unaffected embryo is implanted into the mother's uterus, ensuring that the child is not born with Tay Sachs. Likewise, potential sperm or egg donors can help prevent the possibilities of conceiving a child with Tay Sachs disease. Also, if you don't have any children and you suspect that you might be a carrier because you fall into the ethnicities where Tay Sachs is common, then Tay Sachs can also be diagnosed prenatally through the use of screening such as chorionic villus sampling and amniocentesis. Chorionic villus sampling can be conducted around 11 weeks of pregnancy and involves removing tissue samples from the placenta to check if there is any chromosomal abnormality in a child. Likewise, amniocentesis is a technique to check to see if there is any chromosomal abnormality in a baby by testing the fluid sample surrounding the baby. This test is usually done around 16 weeks of pregnancy. Therefore, this way, carrier parents can take such measures to prevent the risk of having a child with Tay Sachs disease.
Unfortunately, no cure exists for Tay Sachs and the condition is fatal. However, there are some experimental therapies which are not approved by FDA but available in clinical trials which can suppress the symptoms and can increase the lifespan for a short time. These therapies include gene therapy, enzyme replacement therapy, pharmaceutical chaperones, and substrate reduction therapy. There are two different types of gene therapy. In the adeno-associated viral vector gene therapy, modified viral vectors are used to deliver functional HexA genes into the brain tissues of the individual. Since now there are normal HexA gene present in the tissues, enzyme levels are elevated and symptoms of Tay Sachs are alleviated. In a recent research, the use of gene therapy using AAV for increasing the activity of HexA gene has proven to show an increase in the HexA enzymes in mice, which decrease the accumulation of GM2 in the brain and the liver. Now let us look at CRISPR-Cas9. In CRISPR-Cas9, an RNA with a correct copy of the HexA gene associated with the nuclease is inserted into the tissues of the person affected with Tay Sachs. This essentially deletes the mutated copy of the HexA gene and inserts the normal HexA gene into the genome of the individual with Tay Sachs. Since this permanently removes the mutated copy of the HexA gene from the genome of the individual, it means it can be a more permanent treatment solutions to individuals with Tay Sachs. One advantage of CRISPR-Cas9 gene editing is that it's incredibly precise in which segment of DNA or the genome it removes and which segment of DNA it inserts into the genome of the individual. So there are less chances of adverse side effects, such as off-site mutations.
The next treatment method we are talking about is enzyme replacement therapy. In this method, mutated HexA gene is replaced by a normal HexA gene. A recombinant gene can be produced with the help of alpha and beta subunit of HexA gene. By treatment from this method, the GM2 deposition decreases and the activity of brain increases. For effectivity of this method, the enzyme must be able to cross the blood-brain barrier effectively. So in this diagram, we can observe that there is a mutated gene and this is a packed viral vector encoded with the human HexA gene. After a few weeks of the therapy, the mutated gene expression is suppressed and normal HexA gene is expressed. So that we can see here in this diagram, normal genes are expressed. Another treatment method is pharmacological chaperones. Pharmacological chaperones are small molecules that bind to the target protein and promotes the correct folding of protein. Pyrimethamine, a pharmacological chaperone, increases the activity of HexA gene in Tay Sachs disease but does not have any effect on central nervous system. Progranulin, a type of glycoprotein released by different cells, act as a co-chaperone with HexA and decreases the accumulation of GM2 in the fibroblast cells of the Tay Sachs disease. Still further research is done to observe its effect in different stages of disease. Further treatment includes bone marrow transplant, stem cell transplant, and platform technology. But for this treatment, further research is required. In this methods, expected success is not observed, and this can be a future research treatment, but further research is required to get this research into human models.
Having a child with Tay Sachs disease can feel overwhelming for any family. But you're not alone. There are many supportive care available to help families at risk, such as genetic counseling, educational support, financial support, and emotional support. As gene therapy and enzyme replacement therapy are ongoing researches to obtain a cure for this disease, until then, genetic counseling is highly recommended for couples with high risk for Tay Sachs. It is important to get screened before or during pregnancy in order to prevent the disease. First, let's see how genetic counseling works. A counselor helps the parent understand the risk of having a child with a genetic disease. They explain screening options to the parents in order to test if he or she is a carrier of a disease. To analyze this, blood samples are taken to determine the levels of beta hexosaminidase enzyme activity in a parent and therefore detect the presence of a disease. Here, you can see that we have a blood vessel where the black dots indicate the beta hexosaminidase activity. And in this case, we see low levels of this enzyme activity, which indicates the presence of Tay Sachs disease. In addition to this, genome sequence of the parents can also be determined based on the blood sample to find out whether this parent has a mutated HexA gene or not. And therefore, this can help detect the presence of Tay Sachs disease. After performing all these tests, genetic results are obtained. Then a genetic counselor interprets the results, discusses the outcomes of the test, guides the family by providing them alternative options, and therefore, this way, genetic counseling can be a great support in guiding the family, and thus it is highly recommended.
Tay Sachs being a rare disorder and not much information about it, educational supports are a helping hand for the patient and their family as it promotes patient centered treatment. And adherence to medication and treatment, this reduces complication to illness. Also, there are online pretest educational tools available for Ashkenazi Jewish population and carrier screening is also available in Quebec. Sick Kids, Canadian MPA Society, are non-profitable organization to raise funds on behalf of Hospital for Sick Children. National Tay Sachs and Allied Disease of Canada is a NGO that provides education about disease, provides parent-peer support groups to eliminate Tay Sachs in Burlington, Canada. Northern National Tay Sachs and Elite Disease Association being a research institute that provides assistance for medication of rare disease. Jewish Genetic Disease Consortium is a support group and awareness group for the Jewish people with the Tay Sachs as it is the common disease in Jewish population. Thus, Tay-Sachs being a neuronal disorder which is caused in 1 of 100,000 live births and 1 in 300 of the carriers, which is not a quite common disease and there is no permanent cure. But it definitely includes therapies to prevent it and its symptoms. Thus, spreading awareness to support for affected individual is important. For the therapy, genetic screening is an essential step to identify what is the stage of the disorder or any specific changes as DNA level which can help to diagnose, treat and prevent illness. SickKids, National Tay-Sachs Elite Disease Association, and National Human Genome Research Center are effective and non-profitable organizations that together enhance the process of treatment along with the mental and financial support to affected children and their parents. All in all, this supports mentally prepare for what is coming next to avoid complications and smooth treatment. In summary, Tay-Sachs is an autosomal recessive condition that is caused by mutations in HexA gene. There are not any treatments for this disease, but there is a lot of research being done on methods like gene therapy. Please remember that there are options to help you financially and emotionally during this time. Thank you, and I hope our video helped you feel more confident in your knowledge of Tay-Sachs disease.